All right. Let's do, yes.
I know you record the lectures, so we can like watch the
Not my plan. Because I find out that like when I've done that with COVID, like students like turn off during class and they're just like, oh, I'll watch it later. And then it doesn't happen. So Rachel's not here, so I gotta provide it for her. Because she has another class, which is why I'm here. But we will see. Maybe for a review or something. That's a good question. I should share that. All right, so today we're going to talk a lot about science, which is why I had sharing of science. And we're finally going to get to the visual cortex. So we're actually going to see something today. All right, so I can see clearly now. We haven't seen anything yet. We're still in the eye. Right? We haven't even gotten out of the retina. All right. So that is the goal today, is to get to the cortex. I appreciate everybody being patient with me as I learn your names, but I think I've got it. Let me just run through real quick. Mackenzie, Michaela, Annalie, Gibson, Caroline, Carolyn, Micaiah, Ansley, Meg, Shelby, Anya, Abby, Madison, Gray, Rachel, Haley, Paxton, Becca, AJ, Ava, Logan, Kate, Faith, and Matt. Okay, so I will work on that, right? I will not see you for 5 days, which I don't like. You need to make sure that you look over this stuff in the next 5 days. Don't take 5 days off from this, right? We're covering a lot of material in here. We're going kind of quick, okay? Caroline asked about recording the class. I'm doing that for Rachel when she can't be here because of her class conflict. I don't like to provide the lectures to you all because I find that you guys kind of fade out during the class and then you're just like, oh, I'll watch the video later, and then things get busy and that doesn't happen, and then all of a sudden you missed the lecture and you're behind and stuff. What I probably will do is before the, like right before the exam, maybe when we review, I might post the playlist, so if you do have questions and want to go back and look at things, then maybe that would be helpful. But I really need you to take notes in here and stay on top of doing the review points after every class. You guys have been around the block. You know how this works, right? That if you don't do that, it'll be a nightmare when it comes time to study for the exam, right? So do it as you go, please. I beg you. Ask me questions as we go. Good deal? Awesome. Make sure you're doing your lab. I've seen a bunch of you over there. That's super. Make sure that gets done by Monday at midnight so that I can do the stats and organize them, add them to the SPSS, and we'll be ready to go on Tuesday for our stats session. I would like to be done with our stats session by 4, but that means that we all need to get in here, get on laptops, and get moving. If you have SPSS on your laptop, make sure that it works. You can bring your laptop. I'll have a laptop card if you need to borrow a laptop to do SPSS. And I'd like to just be really efficient with our time if we can do that. I know you guys would like to do that as well, and not all that out, okay? We're going to do something, so I'm expecting you guys are okay writing my reports. If you want to be writing your intro and method now, you can be doing that. If you want to write it all after the stats session, you could do that. I mean, I'm giving you enough rope to hang yourself. Either way, you're not turning in drafts of things, right? You guys are the good ones. You're the upper line, right? So you've been around the block, right? Okay. All right, let's do this. What are mock bands? Anyone read about that in the textbook? The what? Just finished praising this. All right. So at point, what do you see? So here we see a gradient from dark gray to light gray. And what do you see at point D and point B? Where the arrows are. Yeah, you see lines, right? It's darker right here and it's lighter right there, right? It's not. It transitioned from dark and went gradually to light without becoming brighter there or darker there. Why would that happen? Why would you perceive that?
Lateral inhibition.
Lateral, thank you, Kate Reeves. Like, I don't know, we spent like an hour talking about this last time. Lateral inhibition, right? So this is all the same, right? This is darker, right? So therefore, there's more inhibition right here, right? From this side. And over here, there's less inhibition from this side, so it appears darker, right? So this is what, if we were to put numbers, this is what happens. Intensity is low, it goes up like this. That's physically what's happening here. And if we were to assign numbers to that, it'd be like 2, 2, 2, 2, 2, 3, 4, 5, 6s, all the same. But what you perceive because of lateral inhibition, if you drew a bunch of receptors, we're not going to do that, or use my practice sheet and diagram this out, what would happen is this, right? You would get a bump down here and a bump up here based on the fact that there's constant more inhibition here and constant less inhibition here, right? So make sure you understand that. That's an important point, right? Also make sure, I'm not going to go back over it, that you understand convergence. It's a key. We're going to talk about convergence and divergence. We're going to talk about divergence today because we're finally going to get to V1 if I shut up and move on. So I'm going to do that. Okay. All right, the binocular pathway. We have two eyeballs. Two eyeballs, and they see some of the same stuff, like the fovea is in both of them, right? And they're overlapping, but some of it is not, right? And so we're going to talk about things, I'm going to turn around and face the wall, right? Like left visual world, right visual world, okay? The left and the right visual world are actually hitting both of my eyeballs. It's not like this eye only sees from here over, right? Okay. Man, was I supposed to do that? Oh, demonstration today. No, I'm not doing that today. Good, I didn't forget anything. I had a moment of doubt there. Okay, so we have two images, right, from each eye. And we have to consolidate those images into a single perception, right? We don't want double vision. That's what, when those images don't get matched up, then we have double vision or diplopia. Right? So the images that hit the nasal part of the medial part of each eyeball, right, match up with the opposite eyes, the contralateral lateral area. So we have to we have to match up the nasal part of my right eye with the lateral part of my left eye, and that has to then match up in the LGN, and vice versa here, okay? All right, so this is what that looks like, right? We have 1 through 9, 1 through 9, 1 through 9 represented on each eyeball, on each retina, okay? Again, the nasal part and the lateral part that are in red here represent my right visual world, have to come together and get consolidated in my left visual cortex. Okay? So we want right visual world, contralateral, right? Most of the senses are contralateral. Right visual world is going to go to my left visual cortex. Left visual world is going to go to my right visual cortex. Okay? The first synapse in the central nervous system is going to be where? In what structure? Yes, the LGN, which is part of what structure? Broader structure. The thalamus, right? The thalamus is going to be your friend in here because you know every sensory system is going to truck through the thalamus on its way to its cortex, right? So, probably good to create a table or chart where you associate which nuclei of the thalamus belong with which sensory system. And so here we are at vision. We're at the LGN, or the lateral geniculate nucleus of the thalamus. Why is this aspect? Okay. Maybe you can hear me better. All right. Now there are secondary pathways. So the LGN is our visual perception that's going to go to our cortex, right? But there are some secondary pathways too that go elsewhere that are subconscious, not perceivable. We don't really perceive them at a conscious level. Depends on what we call perception, right? Brain areas are stimulated, but we're not consciously aware of that perception. And actually 3 different areas. So the superior colliculus is for what? Anyone know?
Balance.
No? Kind of, but no. Related to balance. Yeah, Lex.
Spatial awareness?
Sort of. It's for specific eye tracking or head orientation to visual stimuli. So when something moves in our environment, we kind of automatically, without thinking about it, look at that thing and identify it and sort of identify its movement pathway. And that's happening through the superior colliculus. Inferior colliculus does the same thing for sound, helps orientate our head to our sound. So it is an awareness, a spatial awareness, and it helps with balance. But it's really orientating to visual stimuli at a subconscious level. The pretectum is involved with constriction and dilation of our pupils. You're not aware of that, right? The amount of light coming in automatically causes that through the pretectum area. And then the hypothalamus, what's the job of the hypothalamus in general?
To regulate?
To regulate things. So why would we send visual information to the regulation center? What thing that's regulating our body might use vision?
You know when you are about to say something and you just forget the word on the spot?
Yeah.
Sleep awake and stuff.
Yes, sleep awake stuff. Your sleep wake cycle. Very good. So your circadian rhythms, we send light signals to the suprachiasmatic nucleus to help reset your biological clock. And the most effective way to get over jet lag is to go out in bright sunlight, because that's what is resetting your biological clock. In particular, bright sunlight, not just incandescent fluorescent light. All right. I will point out that prior to the optic chiasm, where it switches over, we refer to these neurons as nerves. There's no synapse. The same neuron, we call it a tract here, because that's sort of the artificial designation of peripheral nervous system and central nervous system. Okay? So pathways, connections of neurons in the central nervous system we call tracts, not nerves. And we call those same things nerves in the peripheral nervous system. All right. Hypothalamus does circadian rhythms. I pointed that out. Superior colliculus does automatic eye movements. The pretectum Enger-Westphal nucleus controls the pupillary light reflex. So light signals come in to the pretectum. The pretectum sends those signals to 2 nuclei, one for each eye, motor outputs, that then go back out and constrict your iris, right? Anyone know what this little triangle thing is? We're sort of underneath the colliculi in the brains.
I'm not sure. Is it the optic?
No, it's not.
Those kids are one of the best.
One of the ventricles, right? Which is the cerebral aqueduct. Looks like a pipe, right? So, when is somebody concerned or interested in your pupillary light reflex? Is anyone ever interested in that?
In concussions.
Huh?
In concussions.
In concussions, right? So if this area of the brain is swelling, you get a blown pupil, right? That's bad, right? You got to get in there and fix things quickly, right? But what else? Who else shines the light in your eye to see if it's...
Optometrists.
Well, not optometrists.
Police.
Police, why do they do that?
Intoxication.
Because drugs, particularly alcohol, circulating through the cerebral aqueduct inhibits this nucleus, and you're not able to do that pupillary constriction appropriately because the alcohol saturates that area. And so that's why police officers use that as a check for intoxication. Right? All right, so now you're right there. The motor output goes back to cranial nerve 3 actually to do that. Okay. Cool. Oh, one last thing. All right, I talked about it. One last cool thing about the plan, that's cool. Interesting thing, I don't know if this is interesting, cool, is in cases where people have cortical blindness, so they're blind because their cortex is damaged or not working correctly, right? There's nothing wrong with their eyes or their optic nerve, but it's a V1 issue, right? These people often have what we call blind sight, where like if you threw a ball at them, they may duck out of the way, and they would say, I don't know why I just moved. I don't have perception you threw a ball at me, but because this is working subconsciously, they can avoid hitting things or avoid walking into walls and things like this. All right. We talked about that. They crossed over about the chiasm. Important thing to note, right, is that your eye is organized retinotopically, the map, we call it retinotopic map, for places in space, right? This place over here always is going to hit like the same part of my retina, right? The 8 over here is always going to hit right here and here. And those two spots are always going to match up here and always come together here. Okay? If you gently like push on your eye like this, it will make those images out of alignment and you'll see diplopia. You'll see two images because you're making them not line up in the right spot, and therefore you perceive two things. Right? All right. Visual cortex, V1, back here at the occipital pole. About 250 million neurons. How many axons are coming in in the optic nerve? That was our convergence. From 126 million receptors down to Haley's 1 million. So now we get to take 1 million and we can blow things up. That's divergence. We can go send those 1 million. They can have lots of different connections. Right? They don't have to go to one place. They can go to lots of places. And that's the divergence part. Okay? Beyond V1, which we also sometimes call striate cortex because it has a stripe appearance because columns alternate from left eye, right eye where they're matching up. Okay? We have extrastriate cortex because there we've already assembled one image, so now it's just a single visual signal. And there we have like higher order things. That's where we start to identify shapes, colors, directional movement. And then beyond that, we get into putting the shapes into an object, right? Recognizing a face as a person, right? We get into these memories. Or what to do with that thing. All right. The thalamus is a filtering station, right? Filtering station. Normally, vision doesn't really adapt that much. Well, I take that back. It does adapt. We'll see some ways in which it adapts today. But unless we do some like specific adaptation exercises, you typically don't adapt. Because when you adapt, if the thalamus starts filtering things out, what does that mean about what you're seeing or perceiving? You're becoming blind, right? I mean, you're not seeing, if the thalamus is like, yeah, that's not important, I'm not going to let that signal through, you don't see, you don't perceive what's not getting through, right? And that can be dangerous if you're driving a car, right? All right, so this is the first influence of top-down processing, right? We can have things like, we can have complex inputs from our visual cortex. We also have inputs from the brain stem in here that are both excitatory and inhibitory, because sometimes we want to inhibit our vision. When do we want to inhibit our vision? signals coming in.
You're sleeping.
Hmm?
When you're sleeping.
When you're sleeping, right? And actually, if you let yourself get sleep deprived, you'll start micro sleeping even though you're awake. And the thalamus kind of shuts off your visual system and you'll start to miss incoming information. Very dangerous when people are driving. 1 car accident per hour in the United States, every hour. So 24 accidents a day occur because of microsleeping and fatigue and issues like that. If you microsleep for less than a second at 60 miles an hour, you can change lanes and go off the road, right? So that's why I said you don't drive sleep, okay? Because this thing can shut down and you just don't see the stuff. Just real quick, God, I can't get off on these tangents. They did this study where all you had to do was look at a box that had a light, and every time the light came on, you just had to press a button. Just press a button, right? People were not sleep deprived, full, good night of sleep. 100% of the time, their reaction time was, I don't know, 500 milliseconds, something like that, right? Very good at it, right? Then they sleep deprived the people, had them do 4 hours of sleep for 5 nights. They missed 400%. Their error rate went up 400% where they didn't press the button, meaning they didn't see the light flash. They were micro-sleeping, did not see the light flash. When they saw the light flash, their reaction time was similar, but they were missing all these stimuli that they previously saw, right? Because this was shutting down and they actually didn't see the light come on. All right. And they didn't know. The worst part about that is they don't know they're missing it. Like when you're micro-sleeping, you're not perceptually aware that you're missing that information. You guys have probably experienced this driving down the road, like, I don't remember that last mile that I drove, right? Yeah? All right, I don't know. But you know when Dr. Pittman's on the road. Okay. Now, so, looking at the LGN, there are 6 layers of the LGN, and they're organized by eye. This is the first place the ipsilateral and contralateral eyes meet up. Okay? So the C layers are contralateral, the I layers are ipsilateral. So point A here is going to be shown in both of these layers. There's going to be cells responding, one from one eye, one from the other eye, and they're next to each other. Same thing with B, same thing with C. It's organized retinotopically in that spatial map of our world. Okay? All right, 6 layers. And they're further organized, and so it's kind of weird. This 1 and 2 is the magnocellular layer. 3, 4, 5, and 6 are the particellular layer. We'll talk about what those do in a sec. But it's kind of weird because it goes contralateral, ipsilateral, and then it kind of switches and does ipsilateral, contralateral, ipsilateral, contralateral. It's kind of weird. Again, I didn't design the system. It's not how I would have built it. Okay? All right. So this area, it's called the particellular area, or P cells. And this area is called the magnocellular layer, or M cells. And those do fundamentally different things. There's also some cells in there called K cells. You don't really know what they do. So even the visual system that we know a whole lot of stuff about, you're like, tell me about it. You're trying to cram it all in my head. But there's still parts of it that we don't know. Like there's these cells there that do something. We don't know. We haven't figured that out yet. All right. P cells are mainly receiving input from our cones, from our focal point, our fovea. They have sort of steady responses, which you would want. You want to, like, I'm looking at something, I want it to stay constant, right? They have smaller center surround receptive fields, which means better acuity, not as much convergence, right, from those. And so they're interested in precision and color and detail because they're getting a lot of input from cones. Okay? The M cells are mainly in our peripheral site, and so there's some cones there. You're using your M cells during this light environment, but there's also a lot of rod input to there as well. And they respond sort of abruptly. They have larger center surround receptive fields because there has to be more convergence because we gave up that for the acuity of the fovea. And they tend to respond to movement. So like, what is your peripheral vision used for? It's like to catch something, and then you put your fovea on it to identify it. That's part of the superior colliculus jump, right? You see something moving, I can't identify that, therefore I'm going to orientate my head and look at it with my fovea and identify it. This is another, these are two other examples. I think one's from your text, this is from your text. I'm not sure where I got that from. But you can kind of see these pathways as they come in and what they sort of then contribute to in the visual cortex. There. I'm not going to go into that too much. All right. The superior colliculus mainly gets input from that M. It sort of branches off of the M because, again, it's looking for things moving to orientate our head. All right? And these both go to the LGN. Layers 1 and 2 for M, layers 3 to 6 for parvocellular. And that is the first time that the visual fields get combined into a single image. And we sort of have a detail in the movement pathway that are then going to V1, the striated cortex, right? In the occipital pole. From V1, it gets handed up to the extrastriated cortex, and we'll talk about what all those other areas do on Tuesday, 5 days from now. All right. Or maybe I'll mention it now, but we'll go more into that. We're going to stick to V1 today. V1, primary input, 250 million neurons, feature detection, right? Feature detection is a common theme throughout the senses. How is the primary cortex organized for every sensory system? It's organized to detect primary features, right? Because we got these electrical signals coming in and we have to reassemble them into our world. And so what are some important features for vision? Lines, angles of lines, right? That's real important. If I were to like deconstruct your face into lines, right, you could do that. Edges. We already talked about how important lateral inhibition is to detect edges and contrast, right? From there, we rebuild the lines, the features. Another important feature is movement, types of movement, okay? We rebuild that into forms and shapes in V2 and V3. We have a special color processing area called V4, and then V5 is special for movement processing, and then we move into the temporal lobe. There are two main pathways out of visual cortex, a ventral pathway into the temporal lobe. What's stored in the temporal lobe? Memories. What do we use memories for? We identify, we take that shape and associate a memory with it. That's called recognition, identification, right? And then we have a dorsal pathway that's going up into the, towards the parietal lobe, and that is our action pathway. It's moving towards our frontal lobe. It's where we have to make decisions and make motor responses to what we're seeing. So temporal pathway, what is it? Dorsal pathway, and what do I do with it? Right? And they talk to each other. What is it helps determine what I do with it, right? All right, so sometimes this gets a little complicated, so I'll try and break it down. We have these center surround receptive fields, right? But they're actually overlapping, right? They're not, so like this rod, these rods can contribute to the surround of this neuron and the surround of that ganglion cell neuron, right? They're not separated border to border, they overlap, okay? And so, again, we could have a single one there, or we could have one here that's going to go and activate both of those, okay? All good, great. All right, so if we put a whole bunch of these receptive fields up here and overlap them, what ends up happening, we need more than that. What ends up happening as we overlap these, right, is that we get all of these single outputs going into the brain, and now we can diverge them, and we can take all of these receptive fields here that have this excitatory center, And if we send them all to the same place in the brain, that becomes a place that responds to a vertical line. If we combine all of those. And if we combine all these, we get a horizontal line. Right? And so what happens is we go from a donut center surround receptive field to a angle of line center surround receptive field in V1. So this, if I'm going to say, I don't think I have that drawing. So if we're saying this, all 1, 2, 3, 4, 5 of those neurons send their input to this particular place in V1, it's going to respond to a line, a vertical line, and it's going to be surrounded by inhibition. Does that make sense? All right, great. You guys are doing well. Again, some important names that you see on those standardized tests. Bugle and weasel. You see bugle and weasel, you're looking for something to do with cats, something to do with vision, something to do with receptive fields. And then you get that multiple choice question right, okay? So bugle and weasel were the first ones to record from visual cells in V1. They also, before that, recorded from cells in the retina and the LGN before they got to the cortex. And they found those receptive fields, the donut center surround receptive fields, I showed you that data last time, in those places. And so they expected to see the same thing in the cortex. So they're in there looking for, putting dots on the screen, searching for, hey, where's this dot center surround receptive field? And they found out that while the LGN was responding, they couldn't find the same signal in V1, so they were kind of confused. This is back in the 50s. We didn't have projectors, computer projectors like this. So how were they presenting this on screen? They were using slides. They were dropping slides into a slide projector that was projecting on here a stimulus, right? And what they found was they dropped a slide in the screen that was going to be a dot. And when this edge went through the receptive field, the neuron fired, but then it quit when the dot was there. And that led them to say, hey, maybe it's lines, and they changed from dots to lines, and they were able to find the receptive fields. And they won the Nobel Prize in Physiology and Medicine in 1981 for that discovery in the 50s. Right? They discovered in 81, they just won the prize there. So they switched from dots to bars of light, and then they were able to now map receptive fields in V1. And a lot of what we know about how V1 is organized came from their work. All right, so if we go back to our vertical neuron, right, receiving all those inputs, when you put a vertical line, it responds a lot. As we start to, so there, you're only in the excitatory, not in the inhibitory. What happens if we rotate that? If we now put that stimulus on there, some of it's in the excitatory, but some of it's in the inhibitory, so it doesn't respond as much. And if we make it perpendicular, we make it the polar opposite of that, it's actually now more inhibited than excited, and it shuts off. So this allows us to generate what we call tuning curves for best stimuli. So you end up having these simple cells that respond best to a stimulus. Here it is vertical. As we move away from that, it responds less. The fact that we have 250 million cells in V1 allows us to do that divergence of those 1 million incoming neurons. I should point out, back on that screen I was talking about you falling asleep, whatever number that was, there's 1 million optic nerve neurons going in, but there's 1.5 million going out of LGN. So we have already added information from our top-down influences into that signal by a half, right? We've added 500,000 neurons that we didn't have going into LGN. All right, so this neuron then would fire most to vertical, and as we get away from vertical, it has a tuning curve. It fires less. Good? All right. Another example of the tuning curve. Good. All right. Yes, you can see these different ones. That's fine. Okay. So Hugh and Weasel found that when they drove the electrode in perpendicular to the cortex, straight down into the cortex, there would be a column of cells in the cortex that all had the same tuning curve. They were all horizontal cells. And if they then picked up and went in a different column, it was a different orientation. And so like if they were to cut across the columns, it started out kind of horizontal, and then the next column was rotated, and the next column was rotated, and then rotated. And so they were able to map this out, and what it turns out is that we get these things called hypercolumns or dominance columns, where you have a collection of columns from the left and the right eye that are the same place in space that respond to every possible orientation of line that can be there. That's how this feature detection happens. Okay? So, to put this together, we have input from the left eye, input from, or two hypercolumns, sorry, two different places, right? Blue place, green place, right? One's coming in from the left eye, one's coming in from the right eye. Together, they make that singular image of that line. That collection of all possible orientations is called a hypercolumn. Hypercolumn has the ocular dominance, one from each eye. Okay? If you were to stain the neurons coming in from one eye and not the other, you can actually see the alternation there. That's why we call it striated cortex, because it looks like stripes, left and right eye. Okay.
I think you just said it. So the black and white, it's like left and right, left and right.
Yes, so they only had staining coming in from one eye, so the dark areas are the other eye. Yes. So this is a collection of 25 that I've overlaid here, right? And those 25, let's just focus on the excitatory part, right? And forget about the inhibitory for a second. So if we do that, these are the 25 centers of those ganglion cells, right? All of these right here send their output to this one. So one neuron coming in can can contribute to lots of different columns. Who's contributing determines what that perception is. So, like this guy doesn't go to that column. I shouldn't use pronouns. This thing doesn't go to this column, right? But it does contribute to this column because an angled line, right, would activate that. This neuron here goes to every single column because no matter how we orientate a line in this receptive field, it's running through the center of the green. So it becomes part of all of these. Do you see how the divergence works now to go from these donuts to contribute to lots of different columns to create line orientations? Right? Okay. So now, right, we rotate, and so now the red is the odd person out. So red doesn't contribute to this one, right? But all these others do contribute to that one. And if we were to keep rotating, right, we're now selecting just the green and the yellow. We'll plus these other gray ones because I don't want to color them all because that would be insane, right? Because you can think about if I had all the connections, all the possible connections going here, what a mess that would look like. But that's the benefit of being able to diverge and send your axons to lots of places. Now, this yellow one is part of, we have a different column. This hypercolumn is centered around this cell and is for every possible line orientation centered around that place. Okay? If we pick up and go to the hypercolumn right next to it, it might be centered around this cell. So now this cell becomes the center. And not only does this yellow neuron contribute to this hypercolumn, but now it's also part of the vertical one of this hypercolumn. So again, like massive spreading of these axons in in the divergence. All right. Simple cells, there are several different types of cells that respond to different stimuli. And so simple cells, again, respond to angles of lines. It's simplest, right? For a particular spot. All right, enough about this. Good. Go on through on it. To make it more complicated, because you know the visual system is going to do that to you, right? Color gets laid on top of this, and it gets laid on top of it in what looks like to us, to the observer, a very disorganized way. And in fact, we call these things blobs because they look like blobs. And it doesn't make a whole lot of sense. You'd think like you have a green for everything there, but no, part of that hypercolumn is red and blue. And so anyway, we're going to kind of compartmentalize things. We'll let the high-end visual researchers try to think about how this works together. We're going to kind of compartmentalize and talk about shape perception different from color perception. Kind of keep it, try and not throw all this stuff on top of each other. Break it all apart a little bit, okay? But I want you to know that these blobs exist, and we'll come back and talk about them in the next exam section when we talk about color. All right. So for each of these neurons, there's a receptive field, and if you get outside that receptive field, it's not going to respond, but anything in that receptive field, it will respond to. Complex cells are like simple cells, but they have a bigger receptive field. So a line at this orientation anywhere in this area would cause a complex cell to fire. As compared to a single cell, it would have to be right there or right there. Like that would be a different column for a simple cell. Good? Okay. So because of that, what happens is if a line is exactly the size of that receptive field, it fires the most. And if it gets outside of that, it gets into the inhibitory part. Right? It surrounds it. So because of that, it allows us complex cells, depending on the size of the receptive field, can detect the size of lines. So now it's not just the angle of the line, but it's how big is the line. And they can also detect movement in particular directions. Actually, strength. So, yeah, complex cells can also do size, but we're going to talk about hypercomplex cells that are going to do all three. So let's just talk about movement right now for that. And the movement is direction specific. So here, if we have a line that's moving downward, it produces the most response. The most inhibition would be a line moving upward. We'll talk about how this happens when we talk about movement in the next section. Okay? We can get into the mechanics of it. And if it's moving left-right, we're not going to get a response. So here, this one, you see, has the biggest response to moving upward in an angle, the least response when it moves downward in an angle. No response in any other way. All right. End stop, or also called hypercomplex cells, respond to size. Jumping on that a little bit. So it's orientated to a angle and length. Okay? So we're going to say, and it makes sense because they're called end stop cells. It measures the end of the line. Okay? Complex cells movements, the direction specific movement. They also can respond best to length. So they're really doing three things. Simple angles. Complex angles and movement. Hypercomplex angles, movement, length. Or end stop. Good? All right. Summary of characteristics of those, right? Center, surround, receptive fields. Now we get to lines, orientations, orientation, movement, length. It's a good table. All right. And so the whole point of this is that this is the process, right, of breaking down a very complex visual world into electrical signals that have to converge, but then can diverge in order to recreate our perception of that complex world. All right. So we don't see angles and lines, right? We see more complex shapes. So we have to move sort of beyond that. So again, I'm taking color out of the equation. We're just looking at like a grayscale image. Okay? So if you were to look at this image, what do you see here? What do you see there? Take your time. I'm going to get some time. You can see something, right? So what do you see? That's like the easy question.
Buildings, skyline.
Buildings, skyline, right? How can you tell which building, which parts of things belong to different buildings?
Different colors.
We're not using color.
We're
grayscale.
Shades of white.
Shades, yeah, okay. And like, how do we know that this building isn't part of this building? Contrast, right? Has different changes in shade, right? All the same shade, right? Little difficult here to tell without top-down influences, right? If I just showed you this, you might think that those two things are connected without seeing this other building that has the same shape, right? So we're constantly using top-down influences to help our bottom-up signaling determine what belongs with what. But spatial frequency, changes in contrast, is an important basic feature that helps us identify different objects. Okay? And we actually have spatial frequency detectors in V1. So it's not just lines, but it's how the lines change that the brain also responds to. Detecting alternating light-dark patterns. So this is the same frequency. High contrast means that there's sharp edges. Low contrast means that it's fading in between. But the light-dark frequency is the same. There's the same number of light and dark cycles in each of those. Good? We understand what contrast is? So low spatial frequency means less changes. So it could be dark, less change. It could be lighter, less change, or really light. High spatial frequency, areas like this building, those squares, these bricks. Probably can't see the bricks because you're too far away, because as we get further away from objects, we lose the ability to see spatial frequency. We lose some of the details, right? High spatial frequency, more details represent smaller areas. Okay? Contrast is the distinction between those two. How distinct is the change that's happening. So this is a nice image because it shows on the left we have low frequency, and then on the right we have high frequency, fast changes. All right? Here we have high contrast. Here we have low contrast. If we have low spatial frequency, we start to lose our perception of that with less contrast. If we have high spatial frequency, we can maintain that perception with less contrast. At some point, we lose the ability to detect it when the contrast gets worse enough. Does that make sense? All right. So there's an interaction between the frequencies. Higher frequency needs less contrast. Lower frequency needs more contrast. All right. Sensory adaptation. We talked a little bit about this. The time course can be very quick. If you're adapting in your V1, it's going to be really a fast adaptation. If you're adapting based on the thalamus. It could be a very long, it could be seconds, on the order of seconds if you're not seeing something, right? Can happen from fatigue of sensory receptors or neurons, right? And when we stimulate part of the cortex with the same stimulus for a long period of time, the neural responsiveness starts to change, and that affects those tuning curves. It effectively, adaptation can shift those tuning curves. Does that make sense? Probably not. That was what I'm saying. Do you understand the words? Okay. All right. Again, a shift in attention or stimulus salience for the thalamus can also cause this, right? So sometimes we can use this illusion, this adaptation, to help understand how things work. And we'll look at that with color because it helps us understand how opponent process works in color. All right, so what I want you to do is stare at this bar, right in the center of this bar. Just keep staring at it. Don't move your eyes. Don't move your head. You have, as you're staring at it, I'm going to talk. You have some lines that are different angles above and below it. And in a second, I'm going to change the slide, and I want you to tell me what your perception of those bars is. What did you perceive? They were slanted and then they righted themselves. Did anyone else see that? Right? You can do this with the slides at home. When you get there, your roommate's going to go, hey, come here, I'll show you. Right? And that's how I'm going to make you all geek out in here if you follow me down that rabbit hole. All right, so they moved. Like when we switched it, they were like the opposite of what these were. They were this way, and then they righted, right? And down here, they were this way, and they righted. That's because staring at this for that period of time, shifted your tuning curves so that when I now showed a vertical line, the opposite orientation was greater until the adaptation went away, which is pretty quick, on the order of less than a second probably, and then you actually saw the real stimulus. All right, so that's one way we can know that we get spatial frequency detectors. So for instance, here, if this is a tuning curve, right, for the vertical stimulus we're going to see in the second one, right? But we're adapting to this stimulus, the orientation like this, right? That neural activity is being stimulated here in the first slide because we're on the angled line, right? And so what happens is, as you keep looking at that, the V1 cells start to have a weakening perception. Their activity gets less. That's adaptation. So it shrinks the ability of those cells to respond. So now, when we show the vertical line, instead of it being like this one is less fatigued than so we perceive it as the opposite rotation until that fatigue goes away and this activity comes back up and we're like, oh, no, it wasn't the angle, it was the vertical line. Now, does it make sense? Okay. All right, look right here in the center. I'm going to take my little pointer away, but keep looking right there. So we are adapting to spatial frequency, right? As you're staring there, the picture's constant. Is your perception changing? It's kind of like fading in and out a little bit. I'm not crazy, right? That's happening to other people, right? All right. Now what do you see? Do you see the lines? All right, like blink your eyes and shake a little bit, right? They're not there, they're much weaker, right? So having adapted to that, right, transfers over when we get to the next stimulus. That pattern of activity is still firing after it's gone. Same thing, let's do it again. Right here, I'll show you a different one. Keep your head still, it really helps to, like, you want to get those stimuli on the same columns in your V1. Now what do you see? Anyone see a checkerboard pattern that then faded away? Because there's no vertical lines here, there's a slight horizontal, but you had the leftover adaptation on top of that. So at first you perceive that checkerboard. Good? All right. I feel like I'm flying through this material. You guys okay? Not a lot of questions. All right. So if this is what we perceive in the real world, which it's not because we don't see it in gray, we see it in color, so it's even more complex, right? But if we just try and break it down into spatial frequency, if I want to say, okay, how do I see this gentleman's face? There are parts of his face that are high frequency, and then there's parts of the face that are low frequency. And so we're breaking this complex image into these bits and pieces. In V1, there's cells that respond like this, and cells that respond like this. And then in V2 and 3, when we're going to make shapes and forms, we merge them back together so that we see something that looks like this. That's how we can take simple features from a complex face, break them down into simple features, but then reassemble them into this perception. So, I kind of lied to you at the beginning. I said that we were listening to I Can See Clearly Now because we were going to talk about actually perceiving things. You really don't perceive stuff in V1. You're really perceiving combinations of things. Like this isn't what you see when you look at this, right? So you're really seeing the combination of all of these things together. So maybe we really haven't gotten to perception yet in the brain at the level of V1, right? So there are simple cells in V1 that respond to low contrast, high contrast, medium contrast, high contrast, all ranges of contrast and all ranges of spatial frequency. Remember, we've got 250 million neurons. We can do a lot of stuff with 250 million neurons. Right? And that's probably an underestimate of how many neurons are in V1. All right. So sort of a summary. What do V1 neurons actually see? So over here, this is a video. No, here's the video. So I'm going to show you a real quick video clip of a researcher. And essentially what they're doing is they're doing work in cats, recording from V1 neurons. They're playing a movie. And so there's a guy's face on the movie and measuring what is that cell seeing. And it's a low frequency cell that's seeing the low frequency components of that. Again, it hasn't been assembled back into that picture of the face yet.
Good grammar and spelling are important, but if you want to write essays that inspire,
more and more we will be able to measure and understand how our brain cells function. Deciphering our consciousness is another question, but the experiments of Dr. Yang Dan have proved that in principle it is possible to read and to understand the brain.
I didn't know that we started this thing again. So this is a digitized movie. We present this to the animal and we record the activity of the visual neurons. And these electrical signals that we recorded from will travel through these cables.
She managed to tap the vision from the brain of a cat and to reconstruct what the cat sees onto a computer screen.
So this is a digitized movie. This particular movie is a short clip from Indiana Jones, I think.
Here we see the two images next to each other. On the left, the image that was shown to the cat. At the right, the image that is tapped out of the cat's brain. The image that was shown to the cat.
All right, one points to that.
On the
So we're looking at a single neuron's receptive field, right? So on that Indiana Jones picture, there's a little box essentially located somewhere on the screen, and that's the only thing that neuron is seeing, right? So as something passes through that box, part of someone's face or a hat or a leg, right, then that one neuron is responding to just what's in its receptive field, which is why it looks like that.
left, the image that was shown to the cat. At the right, the image that is tapped out of the cat's brain.
The picture has a lot of random flickering noise.
Yeah.
That's probably the noise of the actual neurons. Because they sometimes fire spontaneous spikes. We consider those noise, but maybe they reflect something else, maybe the thoughts or something. But we can distinguish that. So when we use all of those, thinking that they represent visual information, this is the reconstructed that we don't know.
It is the first time an image was tapped from a brain. It proves that we can read and understand the grammar of it. The face is recognizable, but I still can't avoid the impression the face has something cat-like.
I don't know about that. It's a little dramatic. So. But the point of that is, why do this research, right? Because, like, a lot of blindness is caused by peripheral issues, either optic nerve, damage to retinas, development of things, right? Where the brain is actually pretty healthy, right? So there's a lot of research like this trying to figure out how can we take a camera and take the electrical signals from a camera and feed them into V1 in the appropriate way to create high spatial frequency, low spatial frequency, lines in the right places. Right? Think about how complex with 250 million cells that process is. It's a lofty goal. I mean, think about what we could do to cure blindness if we could create a device that could talk to the brain in that way. But in order to do that, we need to know what do we need to send the brain in order to code it the right way. All right. What is this a picture of? Pixels. The pixels represent someone said Abraham Lincoln, right? So you got that with that much context. If I say Abraham Lincoln, you're like, yeah, I get that. I can see that, right? So not much contrast, right? You have a very vivid visualization memory of the face of Abraham Lincoln, right? And so your top-down influence is searching for meaning, right? How can I take something that isn't quite giving me the meaning and put meaning with it, right? Which is another way we can get from here to here in perception, right? So, again, simple cells, orientation, but also they're simple cells for frequency. Complex cells, end-stop cells, hypercolumns are all possible combinations of an orientation for a particular receptive field or spatial frequencies for a receptive field. Location, and these columns are tuned. When I say receptive field, that's a place in space. So they're actually location. That hypercolumn represents a location in our environment. And we have two eyes that are contributing to that, right? And all of that leads to these feature detections in V1. All right, last thing I want to talk about today is a little bit about development. A little bit about development of the system. So, I showed you the picture at 13 weeks, and you see these ocular dominance columns. But if we look at the visual cortex at 2 weeks, not a lot of that. Right? So it takes time to encode this stuff, right? And in fact, we'll talk about sort of overall development of all the sensory systems towards the end of the course. After we've covered them all, we'll do a lecture just on development. But when humans are born, and most animals, our visual acuity is really poor. And we have to experience stimuli to encode that. The connections are there, are being formed based on experience. Okay? So, this is all postnatal, after birth timelines. Why does it develop postnatal?
They need stimuli.
Yeah, you need stimuli. And what you don't have any, you're in the dark prenatal, right? So, visual system is one of the least developed systems at birth for humans, definitely, and for most animals. The human visual system doesn't become like adult-like until about 9 months. Babies are 9 months old before they can see like we see. In fact, they do most of their identification through sound and smell for the first couple of months because they really can't see your face. And because they haven't developed the spatial frequency to do that, they don't have these ocular dominance columns, right? So experience is necessary to drive us. One of the ways we can measure this is for cats and get all this stuff done to them. But if we take a kitten right when it's born and we restrict its ability to see with a cone and we put it in an environment where the only thing it can see are horizontal or vertical lines, that it can't see any other line, guess what? Those columns develop, all the other columns don't develop. And there's a critical window of the first 6-8 months in humans, not in cats. Like, I don't know how long it takes in cats. It's much shorter because they don't, imagine a cat can't see for 6 months. But in humans, it takes that amount of time to gain that acuity, right? And so now, when we go look at cells that respond in a cat that's raised in a vertical environment, it's missing cells. It's blind to horizontal lines because it didn't develop the columns to detect horizontal lines because it never saw horizontal lines. And a cat raised in a horizontal environment sees lines close to horizontal, but they're blind to the vertical lines because they never developed the columns. You can search and search and search. They don't have them in there. And if you give them a task where they have to see a vertical line, they act like they're blind to it.
So columns like kind of code as you go for what you're exposed to,
Yes.
rather than just being like natural.
Yes. So it's a weird mix. And you can imagine how complex the system is, so trying to understand it in a dynamic system is difficult, right? But it's sort of like the wiring is there, but what the wiring codes for requires experience. So the columns are there, the wires are there from the receptor fields, the neurons, the connections, but it takes experience to make it all work. Good? Good. All right. Astigmatism, so a misshapen part of the cornea, is something that babies are checked for very early on because that can influence. If you have a part of the cornea that's not letting certain angles through, then that's doing this essentially, right? It's limiting the experience. You may not see particular things, which could later have an effect. And so early on, particularly with astigmatisms, children get glasses, they get, we try to correct those so that proper development can happen, right? And there's been case studies in orphanages, particularly I'm thinking about in Russia and Eastern Europe where the conditions are not good for children, where they were laid on their side in a crib for all their early childhood, and they developed, they saw the lines, the bars on the crib, they didn't see other stimuli, and they had visual problems. They had all sorts of visual problems in that case. So it can happen in humans too. It's very rare, obviously, right? Because we typically have lots of environmental stimuli. All right, there are your review points. I feel like I could have gone slower, but you can have your 10 minutes of class time where I'll be happy to answer questions if you have them. Yes.
I just had a question from last year, actually.
Yes.
With the diagram for LGN, it had 3 cells in the alternating pattern. Is it actually 3 rods per lateral cell?
Probably not. That's wrong. Wow.
It's not really.
It was, sure. Which slide?
It was towards 58, or maybe later, it might be 59.
Yeah, these are just examples. So it's not, this is an easy example. We could have 200 receptors all funneling into one, right? And this is only showing in a line. In reality, right? right? Like that might represent that line through there, but then there's like receptors this way. I mean, all these receptors are all funneling into that one neuronal,
Okay.
right? Yes.
Maybe it's like really much better. It was.
And you're saying like your vision is a unit of one. Yeah.
Okay.
Yeah, and then you just add it all together. Right? And when you do numbers that are close together, like that, the reason I did 5 and 10 is because they're like further apart. And so it's talking about like here, you know, you're getting a lot of canceling out sort of things, so the output looks kind of weird. But yeah, you're doing it.
Set that to your dad and say, that's so